In this lesson, we're going to have a look at the process of spermatogenesis, which is the process of creating sperm or spermatozoa. So in this lesson, we'll primarily be looking simply at this diagram here and explaining A, what is going on, B, what are the names of these structures, and C, what are some of the really important processes that are occurring. So to start off, we have this cell up the top. This is a diploid cell. Now, this cell is known as the sperm mother cell. It is the cell in which all sperm are derived from, and this is the spermatogonia. Now, as I mentioned, and as we can see in the cell itself, it is a diploid cell, meaning that it contains one complete set of chromosomes. Now, this spermatogonia undergoes the process of mitosis. And in mitosis, it produces either more spermatogonia, or it can lead to a spermatogonia which will undergo a period of growth. And if this cell undergoes a period of growth, it remains as a diploid cell. However, its name changes as it undergoes a process of development, and it becomes a primary spermatocyte. What I'm going to start doing now is using colour to differentiate each of these cells at different stages of development. So to start off with the spermatogonia, I'm just going to use yellow here, and I'm going to keep the spermatogonia in yellow. Now this first option in the top left, this one here will stay on the edge of the seminiferous tubule or the outer membrane of the seminiferous tubule. This other spermatogonia, as a result of that process of mitosis, however, if I circle that one now in blue, is what is gradually heading inwards towards the lumen of the seminiferous tubule and eventually developing into that primary spermatocyte. So we can see here that the primary spermatocyte is slightly larger than the spermatogonia, and again, that is because it has undergone that period of growth. Now, it's at this point in time that this primary spermatocyte undergoes the first meiotic division. So it undergoes meiosis 1, and at the end of meiosis 1, it produces what is known as the secondary spermatocyte. Now, the secondary spermatocyte itself is a haploid cell, and we can represent that in the cell itself by writing the letter N and just N, to show that this cell has half of the genetic information of a diploid cell, half the number of chromosomes. So in a human, the diploid number, so 2N, will be equal to 46 chromosomes, and in a haploid cell, it is simply going to be half of that, it will be 23. Now, I'll use blue to represent each of these cells. And what we can do is we can draw some lines here to show that this here is a result of that first meiotic division. Now, after the first meiotic division, meiosis 2 takes place. And meiosis 2 takes place resulting in the production of 4 haploid spermatids. Now, these spermatids, while they're haploid, they are all genetically unique. They have undergone these processes of variation, so things like crossing over during meiosis 1 and 2, and this has given us unique haploid spermatids. Now, following a final process, or I should colour these slightly differently here, so we'll make sure these are all pink. And the reason I'm using these colours is to represent them on another diagram in a second. After we have our 4 spermatids being created, there is one final step. And this is a step or a period of maturation. And during maturation, sperm, or as they're finally known, spermatozoa, shed a lot of their cytoplasm. And after they shed their cytoplasm, they also develop a unique tail. So this final maturation leaves sperm looking how we've seen them previously, and we have 4 unique spermatozoa here. So when we're looking at the process of spermatogenesis, these are not only all the steps that we need to know, maybe I'll highlight here in purple each of the steps involved, but also we need to be familiar with each of the names for different sperm cells along their process of development. So if we have a look down in this image here, we can see quite clearly now that these sperm mother cells or spermatogonia are really close to the external membrane of the seminiferous tubule itself. And as they undergo that growth phase, they produce these primary spermatocytes. And these primary spermatocytes are ever so slightly closer to the lumen of the seminiferous tubule itself. After the primary spermatocytes undergo that first meiotic division, they then form secondary spermatocytes, and there are plenty of secondary spermatocytes here. I'm not colouring every single one in, but I'm highlighting quite a few of them before they form those spermatids. In this diagram, you can see the spermatids are beginning to change their shape ever so slightly in that process of becoming spermatozoa. You can even see the formation or the beginning of a little tail forming there as well. Lastly is the end product, and that is the spermatozoa themselves. We can see the spermatozoa and the tail of the spermatozoa poking out into the lumen. So when we're talking about the seminiferous tubules themselves, this is the outer layer of the seminiferous tubule, and this, really importantly, I'll circle it here and then I'll write it so it's really large, is the lumen. Now on the right-hand side, we can see what is in this image, but in a real seminiferous tubule. So if I was to take a cross-section of this one here, and we have a look almost like a slice of pizza or a piece of pie, we could then do the same thing with this cell here. And so what we're looking at in the center of the cell here is the lumen, and then on the outer side we're looking at that outer layer. And as we go through, primarily we look at spermatogonia, moving into primary spermatocytes before developing a little bit further into those secondary spermatocytes, and then we have a layer here of spermatids, and unfortunately it's a little bit too difficult to see the spermatozoa, but we would expect our spermatozoa to be existing in this area here.